Hello and welcome to my 20-minute wrap-up of the lecture on chapter number 6, which is looking at the cell. Again, for a little bit of catching up, the review for the exam has been published, and we can kind of start going on everything kind of real soon. Again, catchphrase is a lot of phrases. I talked about that in class, and I'll leave that for class. Basically, life has order, evolution, adaptation, regulation, reproduction, response to environment, growth and development, and energy processing. These are all characteristics that every living thing is going to have. Again, for this class here, we need to understand what the parts of the cell are, and so that involves quite a bit of just straight memorization, and then also understanding how those parts regulate inside of those. And so again, we'll look at kind of what goes on the outside of the cell, but again, we cover that a little bit more later. Things that are requirement for life are basically oxygen, water, and the nutrients. We're going to talk a little bit more about this and talking about the chemistry. We need reproduction, energy, conservation, organization, energy, and organelles, which are the small parts of the cell that does stuff. Again, plasma membrane, some DNA. These are all structures that together give us the 7 characteristics of life and also give us the themes that we're talking about. I'm kind of going here real quick. This is a great picture of a cell, of staining, of how we can kind of differentially stain different parts of the cells and kind of look at this. And again, this is a living thing. This is a cell. And when we look at this, it's kind of hard to say. It's like, how does that live? You know, we know the parts of it, but how do those parts come together? And we'll talk about that, a little bit about that during this lecture here. Again, things that are too small to see are basically microscopic. We use the light microscope and start. Some things that we really need to know is magnification, which is the ratio of the object's image to the size to its real size. That is kind of how big does it appear. We can make it look bigger by either getting closer to it, or we can increase the size of that particular item. There's a little Gary Larson cartoon of where our early microbiologists will be looking at petri dishes kind of like this. Resolution is the measure of the clarity of the image and the minimum distance between 2 distinguishable points. The resolution of the human eye is roughly about 0.1 millimeter. In contrast is the visual difference between parts of the sample. Now, hopefully we talked a lot about this during the laboratory, and you'll be okay with that in the laboratory. Again, we need to have a general idea of how big things are. I like to think of myself as a metric person, and I used to be almost 2 meters tall and 100 kilograms, but I'm not that any longer. Some of our neurons that go from our hip down to our big toe are pretty big. Again, a chicken egg could be that, but basically what we're talking about is millimeters, is talking about frog eggs. Micrometers, we're talking about bacteria, E. coli. This is the resolution of the light microscope, and then again, this is the resolution of the electron microscope. We are not going to be seeing these kind of smaller items down here. Light microscopes are pretty good. For the most part, you'll be very lucky in the light microscope if you're able to see a nucleus that we see here, and possibly some mitochondria if you have the proper stain. I'm going to kind of go over this. We went over that. Electron microscope. How we know all this is we use a process called cell fractionization, where we take the cells apart and we separate the organelles from one another and look at their biochemistry. How we do this is we use basically a centrifuge. We take some tissues, we put them in a blender and basically grind them up, making a homogenate. This homogenate we spin using centrifugation, and again, depending on how fast we spin, we can get different things. And so things that are heavy, like whole cells and broken bits of cells and stuff like that, would come out in the first spin, and then things like the ribosomes will come out in later spins. And we'll actually do a laboratory for that a little bit later. And so again, be aware of how homogenization and fractionization helps us kind of do a reductive search of what goes on inside the cells. Again, a very important part of a cell is its membrane. I would say the membrane defines the cell, defines the living portion of the cells. Everything on the inside of the membrane is living, everything on the outside is part of the outside. And so again, this plasma membrane is very important. The fluid inside there is called the cytosol. The chromosomes carry the genes. The ribosomes are responsible for making the proteins. Prokaryotic cells are characterized by kind of what they kind of don't have. They don't have a nucleus. They don't have membrane-bound organelles. Again, their cytoplasm is bound by the plasma membrane, and that's basically all there is. Now, prokaryotes can do a lot, and they can do everything that a eukaryote can do. They just can't do as much. This is what a typical prokaryotic cell would look like. Very simple. No organelles. We have basically nucleoid or DNA region. Again, we will have ribosomes that are responsible for producing proteins. We have probably a cell wall and a capsule and a plasma membrane, and that's about it. These are relatively small, typically about half a micrometer across and typically about a micrometer long. And so they truly are microscopic. The eukaryotic cell, on the other hand, is characterized by having DNA in a membrane-bound organelle, cytoplasm regions between the plasma membrane and the nucleus that is invaginated with things like the Golgi apparatus, etc. So there's lots of stuff there. Eukaryotic cells typically are much larger than prokaryotes. Eukaryotic cells tend to do much more. So the analogy that I gave you in class was like a eukaryotic cell is like a cell phone. A prokaryotic cell is like a like a calculator. Your cell phone does everything, including being a phone. Your calculator can only do one thing, and that is to calculate. But it does that one thing very well. Again, you need to know what a plasma membrane, which is this selective barrier that allows sufficient passage of oxygen, nutrients, and waste surface to the volume of every cell. That's very important. We will cover this a little bit later, but the plasma membrane is made up of phospholipid bilayer. We have an outside and we have an inside, and it's work to move things back and forth across this. Now, this is very small. Again, very small, and we'll talk about that a little bit later. One of the things that we really need to look at is the logistics of carrying out cellular metabolism sets limits on the size of cells. And this is something I really didn't talk about in class that I will before the exam, is that the surface area to volume ratio of the cell is very critical to a cell, of what a cell can do. And so what we see is the surface area increases by a factor of 2 to the nth, the volume increases by a factor of n to the third, and I got that wrong, that's to the n to the second. And kind of what this like is, if we take a small little cube like this, the total surface area is 6 units, the total volume is 1 unit cubed, and the surface to volume ratio is basically, surface area over volume is basically equal to 6. However, when we go up larger, we get that volume, the surface area to volume basically down 1.2. And so what we take things like this, if we take an ice cube, a very large ice cube, and we put it on that ice cube, the surface to volume ratio of a large ice cube is very low like this, and that ice cube will melt a little bit slower. Yet a lot of little ice cubes will melt a lot quicker. And so what we see is that if we get an organism that's made up of many small little cells, we can keep this surface area to volume ratio pretty low. Again, things like the plasmodium that you worked in laboratory work because they're very flat or sheet-like. Again, large, chunky, solid organisms tend to not exist because of that surface ratio. Eukaryotic cell has internal membranes that partition the cells and organelles that do the work. And so this is a typical eukaryotic cell, and I want you to know what those cells are and what those parts are and kind of what they do. This is a typical plant cell. Here's the differences would be the central vacuole, which is a fluid-filled structure that gives solidity to this particular cell, and then we have the cell wall. The nucleus contains the most DNA in a eukaryotic cell, and the ribosomes use up the information from the DNA to help kind of make the proteins, and we should know what the structure is. Again, I'm going to go over, I'm going to leave you to look at this, but basically here's the nucleus, and if we look at here, one point that's really critical is that this nucleus is contiguous with the endoplasmic reticulum, and that these membranes, so these membranes are cycled out in here. This is the rough ER, which is responsible for some structure. In the nucleus we have some pores because we have some large molecules that want to go in we have some large molecules that go out. So again, we need to make things like DNA polymerase need to get into the nucleus, but again, things need to get out, like those very large messenger RNAs that are responsible for making the proteins. I'm not going to get into the lamina too much. The ribosomes are particles made up of ribosomal RNA and protein. We will talk about this a lot more in the production of proteins. A model is to have them look a little bit something like this. This is showing your endoplasmic reticulum in the bound Golgi apparatus. The big critical thing that I want you to get here is this thing that we call the endomembrane system, and that is the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosomes, and plasma membranes are kind of all connected to one another, and it's one big huge kind of conveyor belt. And so what happens is that information from the nuclear envelope actually gets converted into proteins in the endoplasmic reticulum, which then get modified in the Golgi apparatus, which then can leave going into the plasma membrane, or when it goes to these vesicles can actually go back into the nuclear envelope depending on what it is. So it's very critical that we have an understanding of how these all connect together, because a lot of times we look at these and look at them and say, yeah, you know, what do they do? But they are actually all one big system, and I showed you that connection between the nuclear envelope and the endoplasmic reticulum. Know kind of what they do. And so here again is the nuclear envelope. Here's that connection I was talking about. It's hard to see in these three dimensions, but again, if you look at it, these are kind of all contiguous. Look at over here. It's contiguous over here, and essentially these transport vesicles kind of bleb off of the endoplasmic reticulum. So it's the rough ER and then the smooth ER outside of that. Again, what do these do? Smooth ER is responsible for production of lipids. So more of those lipids get moved as they get packaged into there, they get moved on. Again, the metabolism of many carbohydrates occurs there, the detoxification of poisons, and things like muscle cells where we see the stores of calcium in there. The rough ER is bound to the ribosomes, and it can secrete glycoproteins. I'm sorry about that. And it has the transport molecules, and again, this is also a membrane factory for the cells. Those membranes are made there. The Golgi apparatus, we see modification of products of the endoplasmic reticulum. So as these products move from the endoplasmic reticulum through the Golgi apparatus, they will get modified, acylated, various things put on top of those. And again, in the Golgi apparatus, we're seeing kind of the sorting and packaging of materials. And so a cell is not as simple as we once thought it was, just kind of this bag of materials in there. That there is actually stuff going on in the cells, and it's like a little factory. It's responsible for making itself, and it's responsible for making everything that it needs. And again, in multicellular organisms, it's responsible for kind of all the interactions that we see. So there is a lot that's going on in individual cells. Lysosome is important because, again, it contains hydrolytic proteins and responsible for breaking down materials so that it can be recycled in the cell. Phagocytosis is a form of digestion, and we see that in the amoeba. You guys did this amoeba, and phagocytosis typically we see in the introduction of outside materials. Autophagy is basically where things like, again, mitochondria that are kind of worn out and broken down are phagocytized, and then their materials can be recycled by a cell. Again, food vacuoles are formed by phagocytosis. Contractile vacuoles are found in many organisms that help you modulate the amount of water that we have inside of a cell. And then the central vacuoles are found in many mature plant cells, in that they hold organic compounds and water, and they are responsible for kind of giving the shape to the cell. The endomembrane complex here, let's look at it kind of really quick, and I will do kind of a video going through. And what we see is the endomembrane complex. We have information coming out in the form of messenger RNA. This messenger RNA is made inside this material. This material travels through all these, through a vesicle into the Golgi apparatus, and the Golgi apparatus it then travels to the outside of the cell. We're missing that there are many fibers that are in here where these, again, these vesicles will move on the fibers. And again, this is materials that are exiting out the cells. This material can also, if required, go back to the nucleus where it is needed. So again, it's very important that we know what the endomembrane does. Some organelles that are not part of the endomembrane complex would be mitochondria and chloroplast. These are unique systems that do a lot. And actually what we think is that the mitochondria and the chloroplast were actually whole organisms, other prokaryotic organisms that were captured by the eukaryotes and kind of enslaved to do what their bidding. And so, again, we talked a lot about why we think that happens, and we have something that's called the endosymbiotic theory that you need to understand that. Again, here's the mitochondria. Basically, you need to know it's kidney bean shaped. We have an outer membrane, and we have an inner membrane, and then we actually, this organism has its own free ribosomes. These guys here are pretty small. The whole thing itself is roughly about a micrometer, about the size of prokaryotes. Okay. Thylakoids and stroma, again, know the structure of the chloroplast and kind of what they do. We'll go on that. The thylakoids are what actually contain the photosynthetic pigments, and we will cover that a little bit later. The peroxisomes are responsible for breaking down materials. The cytoskeleton is very important, and it's a series of networks of fibers that exist throughout the cytoplasm. That first picture that I showed you showed lots and lots of the cytoskeletal elements. These include microtubules, microfilaments, and intermediate filaments. Again, this is showing you that these microtubules go all throughout the cell and are responsible for giving us the basic structure of the cell. Again, there are motor proteins. So again, motor proteins can attach to a vesicle and scoot along using the hydrolysis of ATP to do that. And so here's a picture of us showing that. And again, this is on a very small level. But again, rather than having diffusion and osmosis move things around the cell, which can happen, but it's not very efficient, eukaryotic organisms kind of use this kind of monorail or this motor system in order to move things around the cell. Again, you need to be able to differentiate between the types of cytoskeletal elements that we have. Table 6.1 is very good for that, and again, you can take a look at that table a little bit later and go over those intermediate filaments. The cytochrome is something that we're understanding kind of more and more. The cytochrome is responsible for organizing basically the arrangement of the chromosomes during cell division, during mitosis. Cilia and flagella are a little bit different. We can understand this. Flagella have this kind of side-to-side motion that allows structures to move. The cilia has a stroke motion. I would consider it more kind of like oars on a boat. And again, when we look at these, it's really kind of interesting is that these flagella have a very unique system or structure that they have, and I should talk about this during the laboratory. And again, this dynein allows these materials. So how flagella move, how flagella go back and forth like this, is that we have these movements, these two structures that are able to move past one another, and that's what gives us our movement. And again, we see this movement as these move past one another. Again, they're connected on top. They have a finite lift, and so one bends inward, one bends outward. Okay, and actin and myosin, we'll talk about this during one of our laboratories, and again, it's also used in cytoplasmic streaming for the chloroplast. We saw this in our Elodia, we saw this in the amoeba, we saw that in the Fusarium, and again, we need to understand kind of how that works. The actin and myosin move past one another in a muscle cell, and we'll describe that in a little more detail. We talked about cytoplasmic streaming. Another thing that we need to talk about is the cell wall, which is the extracellular structure that distinguishes basically plant cells from animal cells. That is very critical. Cell walls are, we see in prokaryotes and fungi and some protists that we have as well. Again, we see a primary cell wall, which is relatively thin and flexible, and when organisms are young, and then this gets built upon as organisms get a little bit older. And so, again, here we have two cells that are next to one another, and we have that primary cell wall, and then we have the secondary cell wall, which is put on top of that. Actually, the extracellular matrix is very important. This is structure showing the extracellular matrix. These materials are excreted outside the cells, but it's very critical because how cells communicate with one another, how cells stick to one another, is dependent on this kind of extracellular matrix. And again, the support functions of the extracellular matrix are support, adhesion, movement, and regulation. And so that's it for my lecture today, and I will go on and give you some more items.